Okay, guys, I think we are going to start the fourth day of our week-long 2019 Red Risk School. Welcome back if you have listened before, or welcome brand new if you are new. My name is Zarana Patel. I am co-director along with Dr. Jeff Willey from Wake Forest of this webinar series. I am discipline lead within the space radiation element at NASA Johnson Space Center. And we are on our fourth presentation, so we're almost done. And we're going through content experts who are discussing risks related to human health during spaceflight. So we are funded by the Translational Research Institute for Space Health, and them and us, Dr. Jeff Willey and I, we're interested in trying to advance really innovative, even if they're high risk, approaches to help us successfully explore space. So whether you're new to NASA research or you're experienced with it, we're targeting academics and entrepreneurs and clinicians who can really take technologies that are out there and advance them into the spaceflight space. So today I'm really excited to introduce Dr. Chris Porada. He's from Wake Forest, and he'll be talking about some novel technologies to mitigate spaceflight radiation. So space radiation is always present. It's an omnipresent environmental stressor in space. Everything you bring or even send ahead of you will be exposed to it. So this includes electronics, food, plastics, and of course, biologicals. So biologicals take a big hit, or can take a big hit from space radiation, because our DNA, can often be a one-hit point of failure. So one hit can cause a mutation that doesn't get repaired, that leads to bigger changes. So with that, I will let Chris do the rest of the talking. Take it, Chris.
Well, thank you very much. Good morning, everyone. So let's see if I can figure out how to get my screen shared here. Okay. Okay, as Rana mentioned, I'm going to be talking today about some of the risks associated with exposure to space radiation, focusing specifically on the hematopoietic and the GI systems, and discussing some novel model systems that we've developed and are using to try to study what these risks may be, and also some technologies to hopefully mitigate these risks and make it safer for astronauts to travel into deep space. So as all of you probably know, with the 50th anniversary of the Apollo missions, NASA has started a new slogan, basically back to the moon and onto Mars, with the goal of sending astronauts to the moon, ideally for a fairly long-term presence as kind of a testing ground before then sending them on to Mars. And all of the experts have more or less agreed that by the 2030s, manned missions to Mars will likely be possible, and astronauts are going to be exposed to this unusual environment, both on the way to Mars and on the surface of Mars. Now, one of the first questions that I always thought about from a logistical standpoint would be, how would we actually get astronauts to Mars and back? And it turns out that a lot of people don't really seem to think that this is going to be a major issue. So you see private companies like Mars One asking for volunteers for one-way trips to go to Mars, thereby bypassing the entire problem with propulsion and fuel and everything to try to figure out how to get astronauts to Mars and back. And it's not only companies like Mars One, we also have entrepreneurs like Elon Musk, who about a year ago at an International Aeronautical Congress presented his entire detailed vision, not only for getting to Mars, but building an entire city on Mars and having a permanent presence there. And if entrepreneurs and private companies don't convince you, I guess who better than Buzz Aldrin, who has actually started his own initiative called Get Your Ass to Mars, where he said that the cost driver for a mission to Mars would actually be the cost of bringing everyone home after a relatively brief stay. And in his vision, he sees that there'll be a program of settlement that will schedule most of the crews that go to Mars to just remain there and establish a permanent settlement. Now, thinking about these things, whether astronauts are going to go to Mars and come back or go and stay on Mars, scientists in multiple different fields have identified that radiation is likely to be the greatest biological risk or hurdle facing these people that are going to be in deep space for a prolonged period of time. Now, space radiation, there we go. is comprised of mainly two different components. There are solar particle events, or solar energetic particles, which are the intermittent release of highly energetic particles that are mostly protons from the sun. These travel at near the speed of light and would arrive at the Earth within only about 10 minutes of an event on the sun. Galactic cosmic rays are high energy protons as well, but also are comprised of high atomic number and energy, or HCE ions, which are atoms stripped of the electron shell, meaning basically just the nucleus is left. Now, the GCR originates outside the solar system. It's likely to come from supernovae in active galactic nuclei. And in contrast to solar particle events or solar energetic particles, galactic cosmic rays are continuous and omnidirectional. Now, to give you an idea of how dangerous this radiation is, or what kind of risk this may pose to the astronauts, current estimates suggest that every single nucleus in every cell of an astronaut's body would likely be hit by a proton once every 3 to 5 days while in deep space, and by an HCE particle once every 1 to 2 months during a mission to Mars. And to give you an idea kind of the magnitude of some of this radiation, between the Apollo 16 and 17 missions, one of the largest solar particle events ever recorded occurred. And it produced radiation levels of sufficient energy that if astronauts had been present in space at that time outside of the magnetosphere of the Earth, they would have absorbed a lethal dose of radiation within only about 10 hours of the event. Now, unfortunately, for NASA and agencies planning trips into deep space, we don't really know a lot about the biological effects of SPE and GCR radiation. And the reason for this, you can see in this picture here, the Earth is this tiny little dot. Basically, on the Earth and near the Earth, SPE and GCR are almost totally deflected by the atmosphere and the magnetosphere that act as a sort of shield and protect us from these radiations. Now, one thing you may wonder is, well, we have astronauts on the ISS, and astronauts have spent 6 months or a year there. Aren't they exposed to radiation, and wouldn't that tell us something? Well, the International Space Station is about 250 miles above the surface of the Earth, which is quite a ways up there and puts it outside the atmosphere. But it doesn't even come close to putting it outside the magnetosphere, which extends about 40,000 miles above the surface of the Earth. Now, one thing I wondered, the moon is certainly quite a long distance away. Well, what about the Apollo astronauts when they went to the moon? Can we get any data from them as to what the risks may be? And it turns out that there are many brilliant scientists and engineers at NASA who were smart enough to realize that they could time the Apollo missions to coincide with the period during the month when the moon would actually fall inside what's known as the Earth's magnetotail. And this is a magnetic tail that basically flows out behind the Earth. And all of the Apollo missions were actually timed so that the astronauts were traveling to the moon at a period when the moon was inside this magnetotail. So the astronauts were actually largely shielded even when they went to the moon. So that may raise the question, you know, if you go to the dentist and you get x-rays or have medical procedures done that involve radiation, normally they give you a lead bib or they do some kind of shielding. Why couldn't we just shield the ship and protect the astronauts in that way? Well, it turns out that standard spacecraft shielding, like thin aluminum as used on the space station, provides protection from most of the solar radiation, which is a good thing. However, the shielding actually makes the problem far worse for high-energy GCR or the HCE ions, because it simply splits these into showers of secondary particles, in effect increasing the dose of radiation that the astronauts would get. So where does this leave us? And I thought this was a good quote from Alvin Drew, who was a NASA mission specialist. He said there is the equivalent of a Mach 1 or a sound barrier that exists in terms of GCR. And until we solve that, we're still in the age of the wooden ships and the canvas sail. Until we get new technologies or new ways of mitigating against cancer-induced death, he felt we were going to be limited to about 1 year in space. This is quite a problem for NASA and other agencies trying to send astronauts to Mars, because if you think about this with existing technologies, it'll take 9 to 12 months just to get to Mars. The astronauts would then have to perform their mission on the surface, which one would assume would probably take at least a couple months. And then, regardless of what Elon Musk and Buzz Aldrin have to say, many of the astronauts probably would like to return to Earth after that. So NASA and TRISH have made it one of their top priorities to try to learn about the risks to humans and exposure to space radiation, and ideally to try to identify effective countermeasures to protect the astronauts and make the mission safe. Now, one question you may wonder, and before I got into this field, I didn't have any clue actually how you would study space radiation on Earth, since, as I mentioned, the magnetosphere largely shields us from that. Unfortunately, NASA has set up a facility known as the NASA Space Radiation Laboratory that's located at Brookhaven National Lab. And here you can see the large heavy ion collider, all of the various smaller facilities, and then this tiny little purple box down here is the NASA Space Radiation Lab. And here you can see an outside picture of the building. And then inside, the beam line where you can actually take your samples and place them. And there's a team of physicists that sit there with an entire array of electromagnets to divert various particles off of the collider and off the accelerators and create exactly kind of the recipe that you would like to try to simulate galactic cosmic ray radiation or solar particle events on Earth, so you can study the effect either on cells or on organisms. Now, the overall goals of our studies were to try to use unique model systems to define what the risks to the human hematopoietic and GI system would be as a result of exposure to space radiation. Then we also wanted to develop and test a dietary countermeasure for its ability to protect or mitigate space radiation-induced damage to these two systems, and ideally identify biomarkers that could then be used to non-invasively diagnose or predict damage that had occurred in astronauts. So you may wonder why, with all the different tissues in the body, we chose to focus only on the hematopoietic and the GI systems. It turns out both of these systems are extremely radiosensitive. Importantly, damage to the hematopoietic system can be life-threatening in a very short period of time due to the need for roughly 1 trillion new cells every day. Similarly, the small intestine is one of the most rapidly renewing tissues in mammals, with your epithelial layer basically turning over every 3 to 5 days. And this is fueled by a very small population of intestinal stem cells. Looking at cancer risk, it turns out that leukemias are one of the most frequent radiogenic cancers. And colorectal cancer, the risk has also been shown to be increased by ionizing radiation. Looking specifically at the leukemias, leukemias have a short enough latency that a leukemia could actually arise and progress sufficiently to compromise a mission, for instance, to Mars. Now, looking at the hematopoietic system, the cells we're going to be focusing on are what you see up at the top here, the HSC, or the long-term engrafting hematopoietic stem cells, or short-term engrafting hematopoietic stem cells. And the reason we chose to focus on this very rare and highly primitive population is because it's the stem cells that have the ability to self-renew and proliferate and then differentiate to give rise to basically every one of the blood cells that you have circulating within your circulation at any given time. As a result of that, we reasoned that damage to even fairly small numbers of stem cells could produce a fairly pronounced biological or deleterious biological effect. Now, it's important to realize that while all of these cells are circulating within the bloodstream, they're actually produced in the stem cells that give rise to them reside within the bone marrow, inside this highly complicated little neighborhood, if you will, known as the microenvironment or the niche, where you have the hematopoietic stem cells being supported by dozens of different cell types, each releasing cytokines and signaling molecules that help to renew the stem cells to self-renew to make sure that you have a supply of stem cells that lasts for your whole life, and also enabling the hematopoietic system to respond to injury or insult or infection, for instance, so that it can rapidly produce and release the types of cells that you need in order to fend that off. And this system is what ensures that the roughly 1 trillion mature blood cells that die every single day are replaced. Since the niche is fairly complicated, we chose to focus on the MSC, or the mesenchymal stem cells. And we chose those because they're one of the key components of the niche. And they're also the stem cell population that gives rise to many of the other differentiated cell types that make up this niche. Now, in our study, our hypotheses were that GCR and SP radiation could have deleterious impact on the hematopoietic system, both directly and indirectly, at the developmental stage of the stem cell or very early progenitors. And we reasoned that genetic lesions in these cells could lead directly to leukemic transformation. Now, since the stem cells are also responsible for giving rise to all of the cells of the immune system, we thought that damaging these early cells could also result in generation of a functionally compromised immune system that would then lack the ability to effectively fight challenges such as pre-leukemic transformation or infection. And we feared as a result of that, that astronauts might actually be doubly at risk due to both the potential for induction of leukemia by radiation-induced mutations and the reduced ability of their immune system to then identify and eliminate malignant cells that arose. So we conducted several sets of experiments. The first of these was to look at what the direct effects on human hematopoietic stem cells would be of simulated space radiation. So we took our human hematopoietic stem cells that we isolated from normal, healthy human donors that were of typical astronaut age, so about 30 to 50 years of age. We brought them to NSRL, and you can see our flasks here sitting in the foam holder in the beamline. And we irradiated them with nothing to control for any effects of transport or flying them on an airplane to BNL from North Carolina. We used gamma radiation as kind of a terrestrial reference point against which we could compare the effects of the space radiation. We used protons at an energy and dose that was similar to what the primary SPE was during one of the larger recorded SPEs. And we used iron ions as a simulation of GCR. We also looked at the effects of sequential proton followed by iron ions. After we irradiated the cells, we then looked at chromosomal aberrations, studied their colony forming potential, performed DNA and RNA analysis, and transplanted these cells into conditioned immunodeficient mice to look at what they would do in vivo. Now, you may ask why we looked at sequential proton and iron, and this was based on a couple different studies. The first of these was early work by Betsy Sutherland at NSRL, who showed that there was a marked enhancement of DNA damage if cells received a sequential exposure to protons and then iron. But interestingly, if the cells were exposed to those same two species of radiation in the reverse order, there was no enhancement of DNA damage. And a more recent publication from Greg Nelson's group actually showed a similar outcome in neurons of the hippocampus. And you may think from a scientific standpoint, this is kind of curious that depending which order you put the ions, you get different effects. But it's important to realize that this is actually what astronauts would see in space based on the fluence of the ions in deep space. If you remember, I said each nucleus would be hit every couple days with a proton, but only about once every 1 or 2 months with one of the heavier HCE ions. So you can imagine that by the time each cell in your body was actually traversed by one of the GCR ions, it would have already been hit multiple times before that with protons. So to look at what some of the direct effects on the hematopoietic stem cells were, we irradiated them, as I said, brought them back to Wake Forest, and then immediately assayed their ability to differentiate and give rise to progenitors of different lineages to see whether radiation may impact the functionality of the stem cells. And you can see, looking at HPP-CFC and CFU gems or mixed colonies, which are the most primitive hematopoietic progenitors, we can see that gamma radiation, proton radiation, iron radiation, and especially sequential proton followed by iron, dramatically decrease the ability of these hematopoietic stem cells to give rise to these fairly primitive colonies. And if we did these same assays looking at myeloid progenitors, erythroid progenitors, megakaryocytic or platelet progenitors, and importantly T-cell progenitors and B-cell progenitors, we saw a similar decrease in the colony-forming potential of the hematopoietic stem cells into each of these lineages, suggesting that exposure to space radiation has fairly marked deleterious effects on the differentiative potential or function of the stem cell. Now, in our second set of experiments, as I mentioned, we took these cells that were exposed with each of the radiation types and then used them to repopulate the hematopoietic system of immunodeficient mice. We allowed these mice to engraft for about 5 months and then analyzed them for the presence of human cell engraftment, the levels of engraftment they had, and then looked at the differentiation and lineage commitment of the human cells within the mice. When we did this, we saw that there were marked changes in the overall level of engraftment within the bone marrow of these mice. Interestingly, exposure to radiation actually led, especially in the case of iron, to higher levels of overall engraftment of human cells within the mice. Now, looking at this slide, if we looked at myeloid versus red blood cells, T cells, and B cells, each of these expressed with a different color, we can see compared to the untreated group, any of the radiation types that we used caused marked changes or shifts or skewing within the lineage differentiation that we observed in vivo within the mice. So this very nicely echoes the results that we had of our in vitro colony forming assays, showing that exposure to space radiation markedly alters the functionality or the ability of the hematopoietic stem cells to differentiate into the progeny that they're supposed to produce. Now, interestingly, when we were euthanizing the mice, one of the mice we noted had a tremendously large spleen. So you can see the untreated spleen here, and the spleen from one of the mice that was exposed, that received human hematopoietic stem cells that had been exposed to iron ions. And the spleen was about 30 times the normal size. When we continued euthanizing the mice and analyzing the rest of the cohorts, we observed that this marked splenomegaly was present in about 5 out of 14 of the mice, and only in mice that had been repopulated with HSC that were exposed to iron ions. Now, when we first presented this data, it's known that mice can spontaneously develop tumors or could potentially have something infectious going on that didn't really relate to the human cells. So the first thing we wanted to do was do flow cytometry on the spleen cells from the mice to make sure that this huge increase in the spleen size was actually due to human cells. And you can see here, looking with an antibody to CD59, which is present on all human cells. In the mice that received human HSC exposed to iron, you can see that the spleen, the vast majority of cells within the spleen are actually human, not mouse. We then did some studies in collaboration with the clinical hematopathology lab here at Wake Forest and found that the spleens from these mice, the cells expressed high levels of human CD3, which is an early T cell marker, CD7, which is a lymphoid marker, terminal deoxynucleotide transferase. And looking here at one of the untransplanted mice as a control, you can see that all of these antibodies are specific. And this led the hematology department here at Wake Forest to conclude that the mice that received human hematopoietic stem cells exposed to iron ions had actually developed T-ALL. We were curious how this may have happened or what was wrong with the DNA that had led to the induction of leukemia in the mice. So we performed whole exome sequencing on the human HSC. And here you can see in this Venn diagram, basically each of the different radiation types are depicted in a different color, looking at single nucleotide variants that were unique to each radiation type, shared by 2 radiation types, or common to all of the different treatments that we gave. And importantly, this was performed on 2 different human donors. Interestingly, when we went back and analyzed where these SNVs occurred, we found that both protons and GCR radiation frequently produced unique single nucleotide variants in genes that were related to hematopoiesis and or had been previously implicated in leukemia. And you can see this is in marked contrast, the protons and the iron here, to induction of SNVs in hematopoiesis or leukemia-related genes in response to gamma radiation. And this is just another Venn diagram showing you that looking at 2 different donors and doing whole exome sequencing, we had almost identical results between the 2 donors. So I mentioned that an important part of hematopoiesis is actually the microenvironment or the niche that drives the process of hematopoiesis. So in addition to looking at the direct effects on the hematopoietic stem cells, we were also curious what effects exposure to space radiation may have on the cells of the microenvironment. So to perform these studies, we took human mesenchymal stem cells, again from the bone marrow of donors that were of typical astronaut age, and we selected based on STRO1, which is a marker for very primitive mesenchymal stem cells. We then exposed them to each of the same radiation paradigms that we had used for the hematopoietic system, and then studied both direct effects on the MSC by doing RNA and protein analysis, and looked to see whether it might be possible that damaging the microenvironment with space radiation could lead to biological bystander effects on the functionality of hematopoietic stem cells. And the bystander effects, the way that we did this was basically to take the mesenchymal stem cells that had been either unirradiated, exposed to gamma, proton, iron, or mixed radiation, and then plating either unirradiated hematopoietic stem cells or hematopoietic stem cells that had received exactly the same radiation type as the MSC together and co-culturing them, waiting about a week, and then taking the hematopoietic cells out and assaying their ability to form colonies like we have done with the isolated HSC to start with. When we did this, you can see, and in each of these, the color coordination is the same. So the blue is set equal to 100%, which is unirradiated HSC and unirradiated MSC. The orange is if we irradiate only the hematopoietic cells. The gray is if we irradiate only the mesenchymal cells, and the yellow is if we irradiated both the HSC and the mesenchymal feeder layer with the radiation type. And this is showing gamma radiation, protons, sequential protons, and iron ions. And we could see that in many cases, exposure to any of these different radiation types had effects both on the HSC, as we had shown before. Also, there were biological bystander effects that if the MSC were treated with these radiation types, it altered their ability to support the hematopoietic cells. And in all of the cases except iron, we saw that there was actually at least an additive effect of irradiating both the hematopoietic cells and the microenvironment. And importantly, as with the HSC, we saw similar results with each of the lineages that we looked at. So the take-home message from these studies was that exposing either HSC or the microenvironment to space radiation produced additive negative effects on hematopoiesis or blood production compared to exposing either of the cell types alone. So we were curious what the mechanism might be whereby this biological bystander effect was occurring by damaging the microenvironment. So we performed protein cytokine arrays on the human mesenchymal stem cells after they had been exposed to each of these radiation types. And you can see here just representative chemiluminescent results from some of these blots. Looking in some more detail, we found that there were alterations in cytokines that were involved in chemoattraction of immune and inflammatory cells. Cytokines, interestingly, that are involved in radioprotection. Several of them were downregulated in response to exposure to radiation. Cytokines that are involved in HSC function were altered by radiation. And cytokines involved in immune cell differentiation and proliferation. And one thing I'd also like to point out to you is that in many cases we saw that there were completely inverse effects depending on the radiation type that we looked at. So for instance, looking at GCSF, gamma radiation downregulated expression of GCSF, but proton and iron upregulated that. And in the case of GM-CSF, you can see that proton and iron actually had completely contradictory effects on that cytokine. So the protein array was very nice, but based on the limitations of the membrane, we were only able to look at 42 proteins at a time. And we wanted a bit more detailed dive into what was actually happening inside the mesenchymal stem cells. So we performed microarray analysis. And you can see here in this heat map, doing a hierarchical clustering analysis after irradiation, you can see each of these colors represents MSC from different donors that were exposed to each of these unique radiation types. And you can see that the hierarchical clustering basically separates each of these samples based on the type of radiation that they were exposed to, showing that our data is highly reproducible across donors. Doing a principal component analysis, we can see the same thing basically. The data sets cluster based on the type of radiation to which the cells were exposed. Interestingly and quite importantly, we were curious whether these effects were fairly transient, so basically just kind of a shock to the cells as a result of radiation, and the cells would then return to normal. So we took the MSC when we got them back to Wake Forest and passaged them for 5 passages in tissue culture, and then isolated RNA and performed the same analysis. And what we can see is that even after 5 passages, the cells still cluster into discrete data sets based on the type of radiation to which they were exposed 5 passages earlier. Going and looking at some pathway analysis, we can see that a whole host of biological processes and processes involved in molecular function were altered by exposure to protons. And interestingly, as we saw in the PCA analysis, even if after 5 passages, most of these changes are still present within the MSC. And with iron, we saw fairly similar results. So in conclusion of this first set of studies, our results to date support the hypothesis we had that the quality and quantity of hematopoietic cells being produced can be altered not only as a result of the HSC themselves being exposed to radiation, but also via indirect biological bystander effects that arise from damage to the microenvironment from this radiation. And we published 2 papers based on these findings, one in leukemia and one recently in a special issue of Stem Cells and Development that focused on the effects of conditions of space travel on stem cell populations. Now, one important caveat that you may have been thinking in the back of your mind watching this is that all of our studies were done taking hematopoietic stem cells, isolating them from the bone marrow, putting them in a tube, shipping them to NSRL, exposing them, bringing them back, and then using them to repopulate mice, which is not really what would happen if astronauts were exposed to radiation. Obviously, their stem cells would be present inside the niches within the bone marrow, and the whole body would be exposed to radiation. So in these newer studies with TRISH, we wanted to ask the question, what would happen if hematopoietic stem cells were exposed to space radiation while they were inside the body? Now, obviously, we'd like to continue studying human hematopoietic stem cells because we feel the translation to the human astronaut would be better than studying the murine system. So the way we got around this, since we obviously can't take humans and expose them to space radiation and see what happens, is that we humanized mice and then used these humanized mice and exposed them to space radiation at NSRL. So it was a similar approach to before, except in this case, we took immunodeficient mice, repopulated them with human hematopoietic stem cells, waited until the hematopoietic system was stably engrafted with the human cells, then brought the mice to NSRL with the human hematopoietic system circulating inside them and exposed them to radiation. And importantly, again, these were cells from adult bone marrow, which I'd like to point out is in contrast to and makes the study somewhat difficult and cumbersome. If you buy humanized NSG mice from Jackson or Taconic, they normally use human cord blood derived stem cells because they give higher levels of engraftment. Unfortunately, cord blood cells are actually fetal cells, and due to the differences in DNA repair mechanisms that exist between fetal and adult cells, we didn't think that that was a valid model for the damage that would occur in astronaut cells. So we took the hard road, I guess, if you will, and decided to try to repopulate the mice ourselves with bone marrow-derived cells. And this flow cytometry is just showing you basically that the mice that we've repopulated using this conditioning protocol are successfully engrafted and express multiple different markers of human hematopoietic cells of different lineages. So I'm happy to report that the first cohort of, I believe it was 72 mice, went to NSRL for exposure to gamma, proton, and iron ions during NSRL 18C. And you can see the mice here inside their holders inside the beamline. And they are currently being analyzed to see what happened to the human hematopoietic system in vivo after exposure to space radiation. Now, changing gears a little bit, at the beginning of the talk, I mentioned we wanted to focus on not only the hematopoietic system, but also the GI system. The GI system, it turns out, is quite a tricky thing if you want to study the human GI system. You can't simply take a mouse and figure out a way to humanize its GI tract. So as part of our TRISH project, we're collaborating with Frederick Zandhausen's group at the University of Arizona, who's developed this fascinating in vitro on-a-chip model system, if you will, known as the HUMIX, which is short for Human Microbial Crosstalk System. And this is essentially a human gut on a chip system that's comprised of intestinal epithelial cells, immune cells, and unique in all of the different gut on a chip systems that exist on the planet at this point is Frederick has figured out a way by putting in a membrane that he can actually also incorporate the microbial components of the gut. So we have a way of studying not only the response of the human epithelium to the radiation, but also what effect the microbiome may have on this response. So the previous was kind of a diagrammatic representation, if you will. This is showing you how Frederick actually realized this and managed to fabricate this gut on a chip system. And this is a fairly neat system because it enables us to basically be continually flowing media through this, or cells, or whatever we would like. The epithelial cells are growing inside this little coiled system, and the bacteria are in here as well. And we have access to sample not only the microbial chamber, but also the media that's flowing over the epithelial cells in real time. So we can be monitoring what's happening to the cells as a result of injury or stimulus. So the first generation HUMIX was quite a feat to accomplish. It actually did exactly as Frederick had predicted. Unfortunately, as you can see from this picture, this is a fairly large device. It was probably the size of a small thermos. So for any of you that have been to NSRL, you would quickly realize this is not really something that would be tractable to performing studies in the beamline at NSRL. So during the first few months of our TRISH project, we managed to successfully miniaturize that thermos size device down to something that's maybe about 4 inches across by 4 inches across, and brought it to NSRL for 18B and showed it to the physicists to see what they had to say about it. And importantly, even after miniaturization, Frederick's group was able to show that if he took human intestinal epithelial cells and grew them in the Humics system, as opposed to using a traditional transwell system that most people use to study the intestine in vitro, the Humics system was much better able to reproduce the physiology of the human intestine, in that the transepithelial electrical resistance was much higher in this system, and he had very good staining for occludin, showing that the epithelium formed tight junctions just like they would inside the body when they were cultured in this system. He also did some transcriptomic, microRNomics, and metabolomics of the epithelial cells grown in the Humics, both in the absence or the presence of microbial species. And he showed that they recapitulated very nicely the transcriptome of normal intestinal tissue, and that by adding in one or more species of bacteria, he could actually dramatically change the genes that were being expressed. Importantly, also, since we wanted to be able to use this device to study the effects of radiation, we had to prove that we could actually grow epithelial cells in this device, expose the device to radiation, and have the correct dosimetry to deliver the dose that we want to the monolayer of epithelial cells that we're growing. So we were able to show, in response to 0.5 gray of radiation, we saw a very nice gamma H2AX response in these epithelial cells growing inside the Humics device. And most of the foci went away within 24 hours, as we would expect, as the DNA was repaired. Now, one thing that Trish had requested of us was that they felt that the Humics was a fascinating device, but since no one had actually used that to study the effects of radiation before, they wanted some kind of a comparator or something that we could use to validate the results that we were going to be getting with the Humics. So reviewers suggested that we actually take wild-type mice to NSRL, expose them to each of the types and doses of radiation that we were proposing to use with the Humics, and euthanize the mice and collect the intestine, and use that as a system to compare to the results from the Humics. So interestingly, we saw both in response to protons and GCR, within the mouse intestine, there was a marked induction of leukocyte infiltration and lymph node swelling. So you can see the normal lymph node here in the unirradiated mouse. And in the gamma, it's really not changed much from the unirradiated, but in response to both the proton and the iron, you can see that the lymph nodes in the intestine are absolutely enormous and filled with lymphocytes. We also saw evidence of vascular congestion and possibly some evidence of bleeding, or extravasation of blood cells, at least, in the groups that were treated with proton and with iron. But we didn't see any of these events in either the unirradiated shams or in the gamma irradiated controls. And this is just a close-up of one of the mice, the intestine from one of the mice that was treated with iron ions. And you can see the marked damage to the intestine here, where we have both vascular congestion and bleeding. We also have evidence of edema within the villi. You can actually see all the fluid filling the villi and kind of swelling it and pushing it apart. We also did some staining looking at Claudin-3, which is a marker for the epithelial barrier, which is critical to preserving normal immune and inflammatory reactivity within the intestine. And you can see that in the sham and in the gamma, we have very nice staining of the villi with Claudin, showing that there's nice tight junctions between all the epithelial cells. In contrast, in the group that received protons, and especially the group that received iron, the intensity of the staining for Claudin is dramatically reduced, and in the case of iron, it's almost completely absent, suggesting that exposure to exposure to GCR ions dramatically negatively impacts the tight junctions within the intestine and likely disrupts this barrier function. Now, I'm happy to report also, so the physicists were impressed with the device at 18B, but unfortunately they pointed out to the group from Arizona, the device still contained a lot of metal, which is really not a good thing if you're going to be putting it inside the beam at NSRL. And also it was still too large to do what we wanted. Frederick and his group went back and redesigned it again. And you can see here as a size comparator, this is the new HuMix chip that contains everything that that huge cooler basically contained, now in a device that's about the size of an SD memory card for a digital camera. So there's actually two devices here. And you can see this was at 18C that we placed these devices inside the beamline for proof of principle and to start getting some data from the system. Now, just in the last couple minutes, I mentioned that one of the goals of the project was also to look at identifying and developing potential countermeasures to mitigate some of this damage that may be occurring to these two tissue systems. So we had shown during one of our earlier studies at NSRL that if we treated hematopoietic stem cells with curcumin, it was able to largely protect against a lot of the effects of both protons and iron ions with respect to the ability of these cells to give rise to normal colony formation. And we found that it was actually effective either before or after exposure to ionizing radiation, suggesting that it may represent a good countermeasure. Unfortunately, for us, and also from the standpoint of developing a countermeasure that would be easy to administer to the astronauts, we wanted something that was water soluble so it could be given as a pill or as a dietary supplement. It turns out curcumin isn't actually water soluble, which would lead to very poor bioavailability. So as part of our TRISH project, we're working with groups at Lawrence Livermore National Lab and UC Davis Comprehensive Cancer Center, who have developed a unique delivery system known as nanolipoprotein particles, which they've shown make it possible basically to package curcumin and render it water soluble so that it's orally available and it has good bioavailability. Importantly, we've also done some studies looking at exposure to curcumin, and in vivo, we've shown that this alters expression or upregulates expression of genes that are involved in DNA repair processes, which provides a possible mechanism for the protective effect that we saw in the studies with the hematopoietic cells. So just to kind of summarize and conclude from what I've shown you, our results to date show that human hematopoiesis is markedly altered from either direct exposure of the HSC themselves or via bystander effects that arise from exposing the marrow microenvironment to Mars mission relevant doses of either protons or iron ions as a model of GCR. Both protons and iron ions induced genetic alterations that were distinct from those induced by gamma radiation, and they appeared to frequently occur in genes that were related to either hematopoiesis or had been linked to leukhemogenesis. SEP and GCR radiation also induced marked alterations and damage to the small intestine that was readily apparent within only 24 hours of exposure. We're currently staining all of the intestinal sections from those mice with antibodies to gamma H2AX, PATM, Musashi, which is a marker of the intestinal stem cell population, proliferating cell nuclear antigen, or Ki67, to look at proliferation of the cells, and tunnel to look at apoptosis within the intestine. We've successfully fabricated the HuMix platform and miniaturized it, and demonstrated that we were able to place this new miniaturized device within the NSRL beamline. Humanized mice and the HuMix devices were exposed to both SEP and GCR during 18C, and they'll be returning for additional ions in 19A. And we've shown that curcumin, which has promise as a countermeasure, can be solubilized by loading it into these NLP particles. And it appears to serve as a countermeasure in both the hematopoietic, and we have some very preliminary data suggesting that it may be protective in the GI system as well. So I would like to finish just by thanking and acknowledging all of the people that have made this project possible. The people at Wake Forest, at the Wake Forest School of Medicine, the group at UC Davis and Lawrence Livermore, the group at University of Arizona, and of course also all of the people at NSRL, without whose help we would never be able to do any of these experiments. And I would also like to just end by thanking the funding that supported this work. So we've had grants from NASA's HRP. We have a grant from Space Biology looking at the concurrent effects of microgravity and radiation to see whether they may have additive effects on leukemogenesis. And of course, we're extremely grateful to TRISH for providing the current support that we have for this project. Thank you very much.
Thank you so much, Chris. That was a very interesting talk. So I have a couple of questions, but I'm going to let Jeff go first.
Okay.
Sure, thanks. Throughout this week, there's been a number of questions related to dietary countermeasures, and so I think this fits very well with that. So I guess a first question which was asked is, can you use your chip, or do you have plans to use your chip to look at other countermeasures related to diet or behavior, like a ketogenic diet, or perhaps fasting, intermittent fasting, as a radio protector?
That's very interesting. I'm not sure exactly what Frederick has in mind with his chip. I know he's not limited only to the interest in curcumin and in radiation. I think he's trying to use it to study a wide variety of processes. I know he's spoken with me about using it to try to study inflammatory bowel disease as well. So I think he's pretty open to using it to study anything, any potential countermeasure or multiple different disease states. Okay,
also, in your HUMEC system, have you evaluated, or do you plan to evaluate human gut cells for compromise by intestinal hyperpermeability?
Yes. Yeah, I know that's one of the main focuses. So it's why we were looking at disruption of the barrier that exists between the epithelial cells. So that's kind of one of the key things that we want to look at in the HUMEC system, is to see whether we have altered permeability, which if that occurred, or what we were reasoning is that at least in IBD, that's one of the main things that happens, is that you have a disruption of the barrier so that the microbiome can kind of mix and get exposed to areas that it shouldn't, so that you develop a hyperimmune or inflammatory state within the intestine. So we're guessing, based on what we've seen so far, at least in the mice, that may be something that's happening and set up kind of a chronic inflammatory state within the gut in response to radiation.
In general, can you just, for the audience who may not be as familiar with the NSRL, clearly you described the NSRL and the utility, but can anyone use it? Is it something that you would need to write a grant to use it? Just in general, just describe the NSRL a little bit more for us, if you could.
Certainly. So my understanding is, in order to use the NSRL, I think you have to actually have approval from NASA or funding from NASA to go and use the facility. I will.
Yeah, I'll just chime in on
I think, yeah, I'm not sure.
If you're funded by a NASA grant, you can definitely have access to NSRL. You just have to put in a proposal we call a beam time proposal. Other agencies and other groups can also have access to NSRL, but that will include payment of the beam time, so per hour. NASA will cover the beam time if you are a funded PI for NASA.
Okay. And if I understand, I think there's definitely an advantage to having going through NASA, because the beam line, if I remember, I think is like $7,000 or $8,000 an hour, isn't it, or something, to reserve the time?
It's not cheap, yeah.
Let's talk about the concentration of curcumin that was used, and how that might equate to how much a human would need to serve as an effective dietary countermeasure.
Okay, well, that's a very good question. So I'm trying to remember what the dose was we used. I guess it would depend on the bioavailability, what dose you could actually get to the intestine. I don't know if we've given a lot of thought, I guess, to exactly what dose would be needed in vivo in order to get the same effect that we had in vitro. One thing I will say, I guess, at least curcumin, since it's basically a component of a spice that's eaten, is quite well tolerated, and it can be taken in huge doses. You can, I think, supplement every day without really any deleterious effect. So, yeah, I don't have a specific answer to that, I guess, as to what kind of dose range we would be looking at, or what the astronauts would have to take on a daily basis in order to provide protection.
Sure. Zorana, would you like to ask some of your questions?
Yes. So we have been discussing this thing called clonal hematopoiesis of indeterminate potential. Are you aware of that, Chris?
Clonal hematopoiesis of indeterminate potential?
Yeah. Basically, where some individuals, or small percent of the population, is already predisposed to mutations in their stem cells. And so we've discussed, if you have an astronaut that is part of this small population, and it's hard to identify if you have it already, and then you fly them, they're exposed to this radiation in space. What do you think about their additional risk? Is there an additional risk of getting things like leukemia in flight and post-flight? But also CHIP, as it's called, has been linked to cardiovascular decrements later on. What do you think about that?
Well, I think that's an interesting problem to try to address, because I know actually at the HRP meeting there was some discussion about this as well, is that in order to have that information, I guess you would have to do genome sequencing on the astronauts, which would seem, at least with the way the ethical things are now, I'm not really sure what NASA would be able to do with that information, because if you think about that, if they sequenced the astronaut and said, well, you have a mutation in this particular gene, so that disqualifies you for flight, I mean, that would be kind of a...
Yeah, I mean, I'm not even talking about crew selection as much as forming the crew of their risk.
Okay.
Do you think, like, for example, your system, if you are evaluating, or are you capable of evaluating cells that are predisposed down the path of
Certainly. Yeah, certainly. So at least with the hematopoietic system, that would be fairly straightforward. If we got hematopoietic stem cells from people that had these mutations that might predispose to cancer, we could certainly study what the effects of mission-relevant doses of radiation would be on cells from those individuals compared to individuals that didn't have the mutations, to see whether that may increase the risk.
Great. So I have a bioengineering background, so I'm really interested in these chips that you guys are developing. How many can you fit in a beam?
So let's see, the most recent one that we did in 18C, I think they put 2 devices into the beamline at a time.
So NSRL has a small beam, which is like 20 centimeters by 20 centimeters. They have the option for a larger beam, 60 centimeters by 60. What you're looking at 2 in a small beam?
Yes. Yeah, so it was in the small beam, and I think we were more or less limited to 2 because that was, I think Jerome had only managed to manufacture 2 by the time we got to 18C. So I think if we used the larger beam, we would obviously be able to put more devices in at a time, and we could probably arrange them so that we could get them a little more tightly packed into the beam.
Cool. And have you guys given any thought to automating the culture of these chips, or are they already automated? Because I ask because I think there's great potential to fly these on the ISS and generate additional data in the microgravity and space radiation environment. So automating will definitely help the chances of getting an experiment to fly.
Okay. I mean, I would think since the system works kind of on a series of small micro pumps and things like that, I would think that it should be fairly easy to automate. I don't think that Frederick and Jerome are frequently going in and manually adding and changing media. I mean, I know there's reservoirs with pumps that are basically circulating the media and feeding the cells. So I think it should be a fairly good way along the path to automation already.
Awesome. Jeff, do you want to take any of the new questions that came up?
Yeah, sure, a couple. So if there is an occurrence of leukemia during spaceflight that is related to radiation or combined challenges, whether it's the environment, the various stressors, isolation, microgravity, whatever. But what is the solution? Like what would be the steps in that particular case, right? Is it possible, to treat that particular individual in the flight that you know of?
Well, I'm not a clinical hematologist, so I should definitely preface my response with that, but I would suppose it would depend on the type of leukemia. I mean, certain leukemias would be responsive to various chemotherapeutics, but I mean, treating someone with chemotherapeutics, the mission itself is already fairly stressful on a physical level. I can't imagine undergoing chemotherapy while you were in space as well. So, I can't imagine it would be an easy thing, certainly. I mean, maybe, I know I had talked with someone about this, like the flight surgeons and things, if, I think it was actually at the TRISH dinner at the HRP meeting, if it was likely that a physician would actually be flying on a prolonged mission like this in case something arose, there would be someone that was clinically capable of diagnosing and trying to administer some kind of treatment. And I don't know if there was any answer given to that. I know they said it was in the works, or there were discussions about how that would happen. But to me, it would seem that you would have to have someone with beyond just basic first aid training in order to be able to take care of some of the things that may be arising.
I think that's a great question, and our talk actually yesterday covered some of the in-flight medical capabilities and some of the challenges. I know there is in the works the development of basically a hemocytometer that can read blood cell counts more comprehensively, but I just want to preface and say that, we haven't actually documented a risk of in-flight leukemia, but we are the possibility of, as Jeff mentioned, all these stressors coming together to potentially propagate that. So, Jeff, do you have any other questions?
I do not at this time, no.
I will add, someone asked about is there any possible way to shield against space radiation? Is it just you need a different type of shielding? You need a lot more of it. I will say that things like lead and water are good. They're not great. They're not going to completely mitigate the effects or shield against it. Lead, of course, is really heavy and it's not really on the table right now. So we're looking at plastics, polyethylene, and water, which are really good. But neutrons, which are part of the space radiation environment, will go through anything. So even if you have the perfect solution to galactic cosmic radiation and solar particle events, you're still going to be exposed to neutrons, and you'll have some radiation dose from that. So no, we have not yet come up with a perfect shielding solution. Not sure that we ever will.
I don't know if either of you saw, I think it was about 2 years ago at the HRP meeting, there was I think he was a grad student or a postdoc that presented that he had come up with a way that you could actually shield the spacecraft on the mission to Mars by using an array of satellites that basically created an artificial magnetosphere around the ship. And he had a huge number of people at his poster talking with him about the thing, and I never saw anything more from that. Do either of you know who that was, or whether that ever went any further?
I don't know about that particular person. I know that there's always been talk of generating a magnetic field around your spacecraft, similar to what the Earth has to protect us on the ground. Mm-hmm. Even if you did that, though, I would still say that you're still going to get neutron exposure. Okay. Yeah. Even on Earth, right, we're getting bombarded daily, constantly with neutrons, and receive some dose from that. Okay. Jeff, do you have anything to add?
No, I don't. You're correct. Okay. I completely agree.
Yeah, I mean, one of the best ways to minimize your space radiation exposure is to get there and get back faster. Because most dose, and by there I mean Mars, most of the dose you're going to experience is going to be in transit, so in deep space. So if you can get to Mars faster, you were provided some kind of shielding by Mars atmosphere, even though it's thin, and you're also provided some shielding by the actual planetary body. So get there faster and come back faster. That will be a good way. All right, guys, I think we're going to wrap up our fourth talk of the week. Thank you so much, Chris.
Thank you.
Tomorrow it's going to be Dr. Kristen Faber from the TRISH Institute. She's going to give you all an overview of the TRISH funding research process. So please dial in for that. Thank you, guys.
Thank you.